This interview is with Dr. Edward Giovannucci, Professor of Nutrition and Epidemiology at the Harvard T.H. Chan School of Public Health. His research focuses on how nutritional, environmental, and genetic factors contribute towards various malignancies, specializing mostly in colorectal, gastrointestinal, and prostate cancers. This work is largely based in the Nurses' Health Study 1 and 2 and the Health Professional Follow-up Study. Edward is also an expert panel member for the World Cancer Research Fund and American Institute of Cancer Research. I hope you enjoyed this interview as much as I did, and thank you so much for listening. So welcome, Ed. It's great to have you on the podcast. We'll kick things off firstly, perhaps with you just taking us through a quick overview of your career so far and how you've got to where you are today, but also why you chose to specialise in the nutritional aspect of cancer research in particular.
Sure, yeah. Very briefly, my background, I went to medical school at the University of Pittsburgh, and after that I studied pathology. In pathology, you do a lot of autopsies and biopsies, and while I found it very interesting, it was a little bit not satisfying because you're usually looking at the end stage of something like cancer, and cancer is already diagnosed. So I became more interested in prevention. So I started actually just as a master's student at the Harvard School of Public Health, and then I really liked epidemiology. So I decided to do a doctorate, like a PhD. And so I didn't initially wasn't interested in nutrition. In fact, my diet during medical school in that time was probably pretty terrible by any standard. But I became intrigued by the notion that diet might be related to cancer. In fact, around the time 1980, doctors Peto and Dahl, Sir Richard Dahl from the UK, came up with a report estimating that as many as 30% of all cancers could be related to diet. And they admitted that the dietary factors were not known, like what could be related to cancer specifically, but just by looking at different rates of cancers in different populations with different diets, they concluded that it's likely that diet had an important role. But there was a lot of unknown, so the range could have been as low as 5% or as high as 30 to 40%, which is actually comparable to smoking. So it sort of intrigued me that diet could be important for cancer. And at the time, Dr. Walter Willett at the Harvard School of Public Health was beginning these large cohort studies, prospective studies, where hundreds of thousands of people are followed over time. And they have like a diet information recorded. And these were basically large epidemiologic studies to look at diet and cancer, as well as other diseases, cardiovascular disease, diabetes. So I became intrigued by the possibility that diet could be related to cancer and that we had an opportunity to discover what might be important. So I started there as first a student and a postdoc and then faculty. And I've been there now for over 3 decades. So I'm probably not moving at this point. So, so yeah, so that's hopefully gives you a little bit of a background of my background. Yeah.
Yeah, absolutely. And just to go, before we delve into the specifics, how would you go about undertaking one of your studies? What data sets do you use? Would you use AI, or is any of it ever wet lab?
Yeah, right. So some people may not know how these studies are done. So I'll give you just the basic setup of how the cohort studies are developed. So for example, the Nurses' Health Study is a study of uh, well, there's actually two nurses' health studies, so I can combine them and say there are over 250,000 women that are followed. So the study, the Nurses' Health Study 1 started in 1976. So this goes way back in time. And some of the women are still being followed, the ones that are still alive. And so the way the studies initially we contact people with questionnaires. These are mailed questionnaires. Now there's a little bit more done, with computers. People can respond online, but the study initiated and still includes a lot of mailed questionnaires. And then so people report a lot of information, like their diet, other lifestyle, physical activity, medical conditions. And then every 2 years they get a follow-up questionnaire. So some have been followed for over 40 years. And then at various points in the studies, we also have collected blood samples, and these are archived in liquid nitrogen, kept at very low temperatures. And we do selective studies, like, it could be genetics, measure proteins, lots of things, metabolites. Um, and so the advantage of the study is that we have all the information questionnaire based and also biomarker based prospectively. So you have for example, some information, let's say, goes back to 1980, and some women, and we also have a study in men, but some of the participants developed cancer, for example, in, let's say, 2010. So that's 30 years later. So it's kind of a unique study that has such long-term follow-up. And we also have repeated measures. So we can see, like, if, for example, if we find a diet is important, we can also say when it's important. So for example, when people let's say someone gets cancer at age 70, and then they might think, like, well, what caused me to get cancer? And usually people think of the past few years, like, what was I doing, last year? But for cancer, it often may be what you were doing 40 years ago that's relevant. Uh, and um, and so one of the nice features of our study, which is relatively unique among cohort studies, is we have the multiple measures, and we go back over 40 years or so. And so we can link various factors to cancer risk. So there's a lot of statistical analyses. And people have, so you might ask, well, like, what do you choose to study? Well, there are lots of investigators involved, and they have their own hypotheses. So it's really pretty open in terms of what people want to study. I focus a lot on diet, not exclusively, but a lot on diet. But other individuals, for example, may be interested in, let's say, for example, environmental pollutants, and which could be measured through the blood samples, for example. So, yeah, so I think that gives a bit of an overview of the makeup of our studies. And now, just to clarify one thing, you mentioned wet lab. And I mean, most of our work would be considered dry lab. We do have the blood specimens. For the most part, we don't do very basic type of research, but we do collaborate with a lot of world experts and different labs. And if there's a biomarker, for example, that seems of interest, we would measure, or we would consider measuring that in our cohorts. Since we have limited blood supplies, and it's some of these are expensive, we study mostly things that are pretty well developed, with a good hypothesis and supportive data, let's say from animal studies or other smaller human studies, before we undertake these assays because we might be measuring, some of these factors in thousands or even 10,000 specimens. So we have to be selective.
Yeah. I mean, we'll go a little bit more into tissue markers later. But yeah, definitely having a data set that's that large and over that large of a time frame, it's definitely invaluable, really. Yeah, so we'll delve into some specifics of your research now, starting off with your research into prostate cancer. And this has evolved mostly around the role of specific antioxidants, and one of these in particular called lycopene, if I'm pronouncing that right. Um, so my question is, where is this carotenoid found in your diet, and what did you find was the significance this has in relation to cancer?
Yeah, right. I think it's pronounced lycopene. At least that's the way I pronounce it. But sometimes the pronunciation in UK and the United States is a little bit different. Uh, but um, but anyway, lycopene is it's a it's a precursor to beta carotene. I mean, most people know beta carotene, which is like a provitamin A. Uh, uh, Lycopene it doesn't have any vitamin A activity, but it's converted in a few steps to beta carotene. It's in large amounts in red fruits and vegetables. So tomatoes, as you might say, are very high in lycopene, and there's some in watermelon and strawberries. So not all of the red foods are due to lycopene, but lycopene is a very common carotenoid. Now, the plant, it doesn't. The plant's not thinking of the human prostate. It's making. It probably makes a lot of lycopene uh, to protect it from sun exposure. So, like it's a pigment that absorbs sunlight and dissipates the energy. So now, just fortuitously, if a man eats a lot of tomatoes, the lycopene gets absorbed, and just for you know who knows why really, but a lot of it winds up in the prostate gland. I think it's just, by chance in a sense how lipids like it gets it's highly lipophilic, so it's carried in oils and fats. So it's just like with the fats going to prostate, lipoproteins, etc. It winds up in the prostate. And it seems to be able to protect against some of the oxidative stress that's inherent in the prostate and many other tissues. And it does seem to protect against prostate cancer. Now, the association has been a little bit controversial, and I think uh, a lot of studies support it, but most a lot of people really want like a randomized trial, which is, where people are randomized. Our studies are observational, in which we do see associations, but sometimes people question whether they're causal associations or they're just a like lycopene might be a marker of something else, but it's not causally protective. Now, one of the challenges is that based on our data, it may take decades to see a benefit. So this is another example where what like maybe like for example what a man is eating in his 20s or 30s may protect against prostate cancer in his 60s or 70s, where prostate cancer often occurs. It doesn't occur too often in younger men. And to do a randomized trial intervention would be almost impossible, or I mean, to do one of 30 or 40 years where you get men to be, blinded, taken lycopene or placebo, and then followed over 30 years. It's just infeasible. So it's really hard to get that kind of definitive data. So we rely a lot on epidemiologic studies and supporting, and there's a lot of animal studies that strikingly support lycopene as a protective factor in prostate carcinogenesis. So these are very good animal models of prostate carcinogenesis, and animals you can manipulate more. Like some only get lycopene and some never touch lycopene, and the ones that get lycopene very consistently get lower prostate cancer. So we, I think it's a likely causal association, but it is hard to prove 100% definitively.
Yeah. With you saying that, what springs to mind is like whether it impacts the epigenetics, because 40 years later, maybe that would be something worth looking into. What do you think?
Yeah, that's a good point. Yeah, and we are planning studies, like we do get another part of our cohort studies is for cancers. We do get the tissue from the different pathology labs. Like these patients are all over the country. It's not like they're our patients in a clinic, but we do get permission from them to acquire the pathology blocks. So when someone has, for example, prostate cancer, usually often they have their prostates removed, and then the pathology lab, looks at does some histology to see what that it is a prostate cancer, for example. But then they keep most of the tissue embedded in paraffin, so it's like pretty stable. So we ask for the specimens, and then we can look at a lot of things like mutations, mRNA expression in the tissue. So yeah, we are doing studies along those lines.
Yeah,
Yeah.
yeah, amazing. Um, so as well as for lycopene, you've studied how different nutritional factors impact the levels of insulin-like growth factors and also insulin itself, and how that can influence cancer development. So with this, what developments have there been towards understanding how diabetes is related to cancer?
Yeah, right. It's been shown for many years. It's actually one of my early interests, even as a student, that diabetes is associated with cancer, at least some cancers. So people who get diabetes seem to be at higher risk for getting some cancers, such as colorectal cancer. Um, now, I actually had hypothesized. This is around 1990, that it's the reason you have that association may be that in type 2 diabetes, which is the most common, and I'm simplifying a lot here, but basically what happens is, let's say individuals gain too much weight or are physically inactive or have, poor diet. These all contribute, and they become insulin resistant, which means that they need higher levels of insulin to maintain their blood glucose. So the cells are less responsive to insulin, but they can still maintain their glucose, but they just produce more and more insulin. Now, that, it's not a good situation, but it's a stable situation until they're not producing enough insulin. The pancreas actually loses ability to produce insulin. And over time, that becomes prediabetes. And then when the situation gets worse and worse, it becomes diabetes. And by the time someone is diabetic, and especially if has diabetes for, let's say, 10 years, they actually, at that point, are actually making less insulin than a normal person. So it's kind of an interesting situation where there's a high level of insulin, and then over time, insulin goes down. What I think is now, I mean, so that basically describes diabetes. Now, I think some cancers, particularly colorectal cancer, and now there's pretty definitive evidence for this. High insulin is the risk factor. So it's not even so much the diabetes itself, but it's the fact that people who have diabetes have had high insulin for years or even decades, and insulin can act as a growth factor. in a simple sense, like hormones like insulin and insulin-like growth factor, they're elevated when there's an excess of like food, energy, protein, and when if you go into like a starvation mode or fasting, insulin, IGF go way down. The levels go way down. It's almost like your body is saying like, well, there's not a lot of food, energy around, so let's, what we're not going to waste time or energy in cell proliferation, particularly in tissues like the gastrointestinal system. So why should the GI system be very active when there's no food around? Um, so anyway, it may just be that this in the state of constant cell proliferation, it just increases the chances of getting a mutation in an abnormal clone and get cancer. So we think it's likely that the high insulin and IGF, also insulin-like growth factor, just promote cancers. They're not like necessarily causing mutations, but they're this constant stimulus of cells. And for example, it's well established for breast cancer. For example, why women are probably 100 times higher risk than men at breast cancer. It's because the estrogen levels, women have higher estrogen levels throughout their lifetime, and that constant proliferation of tissue just by chance over time will cause mutations in some, and some will turn to cancer. So that's like likely why diabetes is related to cancer.
Yeah.
It's more the insulin, yeah.
It's interesting though, because I've read up, I don't know whether this is true, but I've read up that for cancers like prostate cancer, it's actually related to a lower risk.
Yeah, yeah. A prostate cancer is a very enigmatic cancer. And it's also interesting that prostate cancer, um, there, I mean, this is oversimplifying to a degree, but you can think of it as two kinds of prostate cancer. One is fairly benign. They're very common, but they never or really progress. So that's when, like sometimes people say that men die with prostate cancer, not of prostate cancer. For those cancers, that's where it seems somewhat protective. However, for the aggressive kinds of prostate cancer, it may actually increase risk of those. So for the cancers that are fatal. So even for prostate cancer, uh, it probably on overall, it does still more harm than good because those are the cancers you really want to prevent, the ones that are aggressive and fatal. The ones that are indolent and are essentially, it's almost better not to even diagnose them if they're not going to cause harm. So I mean, sometimes they get overdiagnosed because we don't often know which cancers will progress, but um, so, so yeah, so the diabetes for prostate, it's a little bit more of a complicated story, but again, it's probably still harmful for the fatal or the aggressive prostate cancers.
It's definitely interesting, and more research, in my opinion, should go into this. But yeah,
Yeah, yeah, definitely, yeah.
yeah, because I've also read up that there might be a link between the anti-diabetic medication that people are taking and with cancer development that way, as well as the insulin levels that you've already mentioned.
Yeah, right. Yeah, there is some evidence. It's not crystal clear, but there's some evidence that suggests that metformin, which is a common medication used in diabetes, it lowers cancer risk, and it does some things like lower insulin levels, which is consistent with that. So, on the other hand, there's some evidence that uh, people who take insulin, so diabetics who take exogenous insulin might be at higher risk. Some of those studies are hard to tease out causal factors, and like for example, like people who may be taking insulin, well, they're the ones who are maybe producing very low levels of endogenous or their own insulin. So it's a little bit hard to tease out what's going on, but there is some evidence supporting that. Yeah.
Yeah. Yeah. So along this theme of factors impacting insulin levels and IGF levels, we'll now go into your work on colorectal cancer. So you've helped to establish risk factors that contribute towards colorectal cancers and have demonstrated how some of these may act through this very mechanism of influencing the insulin levels. So for the listeners, what are some of these risk factors and how might they impact different age groups and different ethnicities in particular?
Yes. Right. So colorectal cancer, I think, is the cancer which is most related to lifestyle factors, diet, things like that. Obviously, there's like smoking is very important for lung cancer and some other cancers. Colorectal is interesting because it has a lot of risk factors. So essentially, what increases risk, and these are, most likely causal associations. It's obesity, uh, physical inactivity, sedentary lifestyle, smoking, uh, excess alcohol, usually more than 2 drinks a day, um, and poor diet, uh, which I'll maybe explain a little bit later what I mean by poor diet. But then there are also some factors such as like calcium or even dairy products which are high in calcium are protective against colon cancer. And aspirin is a factor that's definitely protective against colorectal cancer. So it has a lot of factors, and most of them are, similar to what you would see for diseases like diabetes and cardiovascular disease. That, the good, lifestyle factors impact colorectal cancer in a beneficial way. So most colorectal cancer is preventable. It's the second most common cancer in a lot of populations, usually second or third. And it's high in both men and women. And it could be prevented by screening, but screening won't prevent all of them. So if we find that people that follow the screening guidelines and have a good lifestyle, probably 90% can be prevented. It should be a rare cancer, but it's a common cancer. So yeah, so I'd be happy to go into specific. I think that gives an overview, but I'd be happy to give if you want to delve into some of the specifics more.
Yeah, definitely. Because from the risk factors that you've mentioned, you've investigated how they might influence different tissue markers, as you mentioned before as well, and therefore the tumorigenesis. So for obesity, for example, it's a state of chronic inflammation of the adipose tissue. So you'd expect to see markers like C-peptide or amentin and ones like IL-6, like different cytokines. So through the process of identifying these different tissue markers, have you come across any potential therapeutic targets? And would these therefore be a more personalized treatment towards the person, like the person's lifestyle? Like, could you see that someone has obesity and therefore might have this specific marker and therefore a specific drug might be better for them?
Yeah, sure. Well, um, we'll start with the markers, which you can measure in the blood, such that as you mentioned some of them. So C-peptide is a marker of insulin secretion, and then there's like CRP, C-reactive protein, which is inflammation. Then there's IGF-1. All of these have been associated with colorectal cancer. So if you measure if you get a blood sample and measure, like insulin or inflammation or IGF-1, follow individuals, 10 years later, they'll be at higher risk for colorectal cancer. Now, the well, I shouldn't say easiest, but uh, these are interestingly, they're all modifiable by lifestyle. So for example, like as you mentioned, body weight is important. Now, individuals, I realize some individuals, it's difficult to control body weight for lots of people and their, genetic susceptibilities for obesity. Um, However, regardless or independent of body weight, uh, diet and physical activity, even just walking a bit, all lower insulin and inflammation. So that you can start by these simple things. Now, if someone wants to go all out and run marathons, that's great. But you don't have to go to that level to get benefits. You can walk, you know, start with 30 minutes a day or, every other day if you're doing nothing. That will have a significant benefit. Um, and then diet. I think a lot of the the diet, how diet influences cancer, which I think that's when we started talking, that was my first interest in going into this field. Um, I think it's true these like processes such as inflammation and insulin. Now, when people sometimes people think, well, diet and cancer, is it like what should I eat? Should it be like we talked about tomatoes and lycopene? There may be some cases where like individual micronutrients may be important, but when you look at something like insulin or inflammation, you really have to look at the whole dietary pattern. And it's the composite of what someone is eating. It's not just one food, it's also what someone is not eating. Like you don't want to eat too many processed foods high in fat and sugar. All these contribute. So if you look at the composite diet, you find some people, because of their diet, have very low insulin levels, and some people have very high insulin levels. In fact, if someone is overweight or obese, the diet is even more important because that person is insulin resistant to begin with. So what they eat is going to make a big impact. So, so that's how diet will affect things like insulin and inflammation, and that has a very, very big impact. Now, for drugs, it's a little bit tricky. I mean, if someone is not diabetic, for example, let's say someone's pre-diabetic, might have high insulin levels. Um, you have to be a little careful about drugs. There are side effects. Um, and cancers, even though we say cancers like colorectal cancer are relatively common, and they are. I mean, the average lifetime risk, like without screening and things like that, could be 3 to 5% your lifetime risk, which is, pretty significant. But, in a given year, your risk, might be like 1 in 1,000 or something like that in that ballpark, in that range. So, if you give a chemo prevention, like a drug that protects against that cancer, it has to have almost like no side effects or very low side effects. A good example is aspirin, which is anti-inflammatory and has been definitely shown even in randomized trials to lower risk of colorectal cancer. So if you wanted the name of a drug that will definitely lower your risk, an aspirin will do it. Um, Now, but first of all, you have to be using aspirin about 5 to 10 years to start getting the benefit. Because if you just like if a 60-year-old, for example, starts today taking aspirin, that person may already have some lesions that are well on their way to being a cancer. So if they get diagnosed, let's say at age 65, the aspirin was probably started too late. So first of all, you have to start kind of early, and then aspirin has side effects, like
Yeah.
it thins the blood and causes bleeding. So I would say that maybe um, high risk people. I mean, there's some evidence that people like, for example, have a genetic history, family history of colorectal cancer. Uh, they actually it may be worth for them to take aspirin, but maybe for a low risk average, the low risk person, the side effects might be worse than the preventative effects on colon cancer. So I would definitely start more with the lifestyle and like lots of things help. Like if you drink too much alcohol, you should reduce it or eliminate it. Uh, Smoking definitely is a risk factor. And then things like walking, running, whatever, and just moving about, not being too sedentary. We find like people who spend a lot of time watching television are at higher risk of colon cancer. And it's probably not because of the television, but because they're sitting down. Um, so there are like all these, a lot of behaviors, even, I mean, weight loss would be ideal, but even, if someone thinks it's too hard to reduce their weight, okay, that's fine, but you can still improve your diet and uh, uh, you know so yeah, so I think that's the way I would probably answer that. But we like you talked about precision, and the other way that this could be used is like we know people that are definitely at high risk because of their genetics, family history, and their lifestyle. Um, and those people probably, I mean, beyond, maybe trying to improve their lifestyle, they should start screening earlier. So I think some of these factors are also important in helping identify people who are at high risk. And we have shown that probably there could be 10 to 15 year range in screening starting time based on lifestyle. So in other words, like uh, I'm not making these numbers firm, just to give you an example. Like someone with very high risk profile should start screening at age 40 to 45. Someone with a low risk profile can start at 55, let's say.
Mhm.
Um, because that would make the screening much more efficient.
Absolutely. So you mentioned micronutrients, and you've studied the efficacy of potential protective micronutrients against this impact that these risk factors have. So what are some specific micronutrients, and perhaps through what signaling pathways might they work?
Sure. Yeah, there's a lot of interest, and there are like there are there's a great unknown of like micronutrients that may be beneficial. So for example, there's evidence that coffee uh, consumption, drinking coffee, has some preventative effects on some cancers. Now, coffee, I mean, people usually think of caffeine, but it's even the decaffeinated coffee has the same benefits. Coffee has hundreds of compounds. A lot of them are strong antioxidants. In fact, if you look at, there were some in vitro studies to measure antioxidants. And now there are some questions how relevant these are in vivo, like in people. But nonetheless, let's say these studies of antioxidant capacity, coffee uh, accounts for about half of the antioxidants of the that in our diet. So, people say eat lots of fruits and vegetables. for antioxidants, and that's fine. I think that's true. But coffee itself, but probably because it's consumed a lot, because some people hardly consume any fruits and vegetables, but they may have 4 cups of coffee a day. So they're getting lots of antioxidants and other compounds. So there's some things that we haven't characterized yet. But in terms of the micronutrients that we know something about, um, I would say three that I think have the strongest evidence, at least I based a lot of evidence on more on human studies and not I mean animal studies are very important and offer a lot of insight to mechanisms, but I think it's important to see what happens in humans. I think vitamin D um, uh, is important, and it's been somewhat controversial, but it's kind of been a very interesting finding that we've had now that we've done randomized trials, and that it seems that it doesn't having more vitamin D isn't strong in protecting against getting the cancer incidence, but people who are taking vitamin D have better survival. So somehow their cancers are less aggressive or progress less frequently. So, for example, if you've been taking vitamin D and then get diagnosed with cancer, you're more likely to have a localized cancer than an advanced metastatic cancer. So, vitamin D has been shown to act on so many pathways. It's hard to even know which one is relevant. But I think the ones related to metastasis or progression of cancer is where vitamin D is acting. And so, I mean, definitely more work needs to be done. But, um, you know, people like we did one randomized trial in Boston, um, and people taking 2,000 international units of vitamin D over 5 or 6 years had about a 20% lower total cancer mortality, which is pretty impressive because that's like total cancer mortality. It's not just one cancer; it's broad. I mean, it still needs more work, but it's been fairly consistent in randomized trials. There's never enough. You always want more data, but I think there's enough evidence that vitamin D, as you would say, it protects against cancer mortality. And you do get the benefit relatively quickly within a few years. It seems like 2 years. When we did the survival curves, that they were similar in the first 2 years after people were taking vitamin D, but then they started diverging where survival from
Right.
cancer was lower. Um, another, I think I mentioned this earlier, calcium. I'd say the evidence to me is pretty almost overwhelming at this point that it helps protect against colon cancer.
Mm.
Uh, and so, and some people get calcium a lot from dairy products, milk. Green leafy vegetables are a source too. Yogurt is a good source. Um, but for people that don't get enough, a calcium supplement does as well. So people take calcium supplements have a lower risk, about a 30% lower risk. Um, and the final nutrient I'll mention is a lot of the B vitamins, particularly folate or folic acid, are actually important in DNA synthesis and DNA repair. That they actually, eh, like they're important in the creating the molecules that go into DNA. So, like if you're deficient, you may have things start happening, like you have uracil getting incorporated to DNA, and it gets, repaired, and you might get some double strand breaks, things like at the DNA maintenance and repair. Now, what's interesting is that now, like short-term randomized trials that gave people folate have not seen a benefit on cancer, but these studies go about 5 years in duration. Now, for things in carcinogenesis and cancer progression, initiation and progression, usually like the initial steps, you have damage to DNA, and then some over many years or decades, some of the particular mutations cause clones that can progress ultimately to cancer in some people. But the initial like damaging DNA, so something where folate might act is at early stages of cancer. And so for colorectal cancer, we see that you get the benefit of folate only 20 years or so before the cancer. So if you want to prevent a cancer at age 60, you probably have to be taking higher folate at age 40. So again, this is very hard to demonstrate in a randomized intervention study. But it's also interesting that when we have, and I think what indirectly supports this concept is there are some types of studies, like when people get excessive radiation. Like there have been lots of studies of the atomic bomb survivors and from Japan, and there in fact like the exact second someone was exposed and when in their life they were exposed. And these people, it's well known that they're at higher risk of some cancers, but some cancers don't show up for like 30, 40 years.
Mm.
So, the childhood exposure could cause a cancer in late adulthood. Some cancers, like leukemias, tend to occur more quickly. If you're going to get a leukemia, it could be within, 5 years. But also, that's another example where, you know, we think it's likely the same for diet, uh, that, like diet can impact like DNA synthesis, repair, those factors. And if that's true, then the benefits might be decades for some factors. Now, it's interesting that I talked about insulin, IGF, obesity. Some of those things may be acting late. So I don't want to imply that all the diet factors are occurring, way early in life. And there are some things that even, like that you can do, like improving your dietary pattern, where an older person can start getting benefits later in life. So anyway, in summary, vitamin D, calcium, and folate, I think are three of the things that I think are more solid micronutrients and related to cancer.
Yeah. And of course, the vitamin D and the calcium would go two and two together, really, because of the signaling pathways that D3 has in particular.
Exactly. Yeah. Right. I didn't get into that, but it is very interesting that you know it, the two of the strongest factors are related, and they're also related in the sense of, at least in some countries like the US, the vitamin D is fortified in dairy products. So people who eat dairy products, get both vitamin D and calcium. So that's a good source. And that's, I think, one of the reasons why dairy products are pretty strongly protective against colorectal cancer.
Amazing. So to finish off, just for time purposes, what would be your advice to the listeners as to who to believe when it comes to potential risk factors? Because you hear so many contradictory arguments, and it's hard to know, what to believe. Should you or should you not be doing something? Like, eating red meat, for example, there's a lot of different contradictory ideas coming that way. Yeah, so what would you be your advice for someone like that?
Yeah, well, f for cancer specifically, I have a very easy answer. It's you can look at the World Cancer Research Fund, WCRF, which is based in London. I mean, they're internationally oriented. You know, their their recommendations are it's international. Um, I had the pleasure actually of being on their panel, expert review panel, for almost 10 years until recently. And I still do some, work with them. So I get to visit London once or twice a year for their panels. And they're amazing in that they review the data very carefully. So everything that if you go to their website, um, it's based on evidence and it's based on uh, like synthesis. All of the studies are essentially reviewed and then they're synthesized, and then you have a diverse group of experts with specified criteria evaluating the evidence. And, of course, it's they're still human beings. So there's some people might say, well, that the consensus panels could be biased, and sure, but an individual person, I mean, at least you have multiple people and with different disciplines. So, and I think that the recommendations are relatively conservative in a sense that they don't go overboard, and the evidence has to be pretty strong, and most people on the panel have to be convinced. So if you look at their, evidence that, and they have a summary of like what you should do and what cancers will be prevented, and I think that's a good source. And I shouldn't mean to imply that they're the only good source, but that's an example where, like for cancer and diet, and they're the first place that I would go to. There are others. American Cancer Society is a good source of data, but uh, the WCRF focuses exclusively on diet, nutrition, and cancer. So that's, I think, the most comprehensive. And you can go like you can get over like they have like 10 recommendations, and they're kind of like the 10 commandments, but they're more like recommendations of what you should do, like how much red meat you should eat, or what fruits and vegetables to eat. And then you can also go, like, if you're particularly interested in a specific cancer, colorectal cancer, breast cancer, you can go into great detail. You can link to websites that present all the data and the reasons why the conclusions were made. And they're meant to be, I mean, some of it is for scientists and they go into detail, but it's also meant for, you know, non-scientists to be able to read and get a good sense of what the data say. So that's my answer for that. Uh,
Yeah, I think I definitely will be fact checking what I hear in the media and online.
yeah, right, exactly. And the thing is that the uh, organizations such as that, they look at all the data over many years accumulated up to that point. Now, the media, um, they focus on novelty. So, so for example, if there's 20 studies that shows, something, and then let's say there's one study that seems to contradict it. Well, that's the novel study, right? That's what you want to, oh, look, what they've been saying is wrong. And that seems novel, but you still want to maybe look back and say, well, maybe I should believe the 20 instead of the one. And in most cases, maybe the 20 studies were wrong, and somebody finally got it right. But so there's it's not easy, but I think in general, we always want to look at all of the data and not just, what was published yesterday. So I think that's why I would go more on these large body consensus than a single media report.
Yeah, for sure. Um, so finally, not wanting to open a can of worms or anything, but what would be something that you are particularly excited for in the future in your field?
Um, yeah, there's I, I still like somehow focus on more of the basic things like, diet, physical activity, and uh things like that. Um, Now there are lots of new uh, like omics, like proteomics, where you measure thousands of proteins, or metabolomics, hundreds of metabolites, and there's lots of so many things that can be measured. And when I started research, like we measured one thing at a time. I wrote a grant once to look at one genetic polymorphism. I got a lot of money, got a big grant, collected about 15,000 blood specimens. And I mean, today that would be laughable because you can measure whole genomes and, much cheaper. We do get inundated with lots of data. So I have lots of students and postdocs that know a lot more than I do about a lot of the specifics of these data sets. And, it's like big data, I guess another term for that. And it's exciting to see how these will be used. I still like think that we need hypotheses and ways to synthesize the data and still eventually come down to single conclusions and factors. And not sometimes you show, well, we found 160 metabolites related to this cancer. And so it gets a little bit overwhelming to some degree, but I'm interested in still maintaining a big picture and helping people synthesize all this data. And for example, how this data could be used to better understand basic things like diet. So diet may affect all these metabolites and proteins in hundreds of different ways, but at the end of the day, what will be the final message? And so to find very addressable questions, it's great to use the big data, AI, to help us organize the data, but still to come down to relatively simple, important questions that provide, important information for people.
Absolutely. Yeah, and AI probably holds a lot of power for the future.
Yeah, it's something I'm just learning, scratching the surface, and a lot of my students are well ahead of me on understanding a lot of this. It's a lot. There's a lot of excitement, but we still don't want to lose the forest for the trees and leaves. So there's a lot to synthesize.
Well, thank you so much for joining us today and taking your time out to speak to me and the listeners. It's been wonderful. Thank you so much.
Thank you. Yeah, thanks for listening. Yeah, I enjoyed it. So.